Hi everyone, welcome to our science talk for tonight. We're so excited to have so many of you with us for Brian's talk. So just to introduce those of you who are new to LumCon Science Talks, you can connect with us on this side of the talk by using the question box, and the question box is in your dashboard. So feel free to reach out and make comments, ask questions, whatever you need to do, and use the question box for it. Sorry, guys, I'm really tired today, which will be reflected in my jokes. So to practice using the question box, I asked 2 jokes at the beginning of each talk. So Brian, Craig, are you ready?
I'm never ready. I don't know why you asked me if I'm ready. I'm almost never ready for this.
It's just a good segue. So Brian, in your honor, I have kind of a chemistry related joke here.
Oh boy.
Oh boy, you ready?
Sure.
What did the thermometer say to the graduated cylinder? Audience, if you have answers, just type them in your question box.
Why do I always start laughing before you give the punchline? Because it's just like the anticipation of how bad it's going to be.
Do you know the answer?
No, I have no idea what the answer is.
Oh, ooh, our audience has some good ones. All right, Brian, do you have an answer?
No.
No. The thermometer said, "You may have graduated, but I have many degrees." Oh, come on, you guys. Just giggle a little. Brian's never had a speaker walk away. Yes. Reached the new height. All right, this one is for you, Craig.
Oh, great.
What do you call an alligator in a vest?
No idea.
Oh, you have some good ones, audience. An investigator. Good job, Tristan. Tristan got that one.
Oh no, Mert.
I think they're funny.
I don't know how you manage to both find the jokes and then always like top, but I don't know if top is the right word. The jokes from the last time.
Find new levels.
That's right. Find new levels.
Some talent can be learned, and some talent is just inherent. And on that note, I'm gonna turn it over to you.
Thanks. Thank you, Mert. Appreciate it. It's always hard to follow that, Brian.
Yeah.
Yeah. Where do you go from that? Well, good evening, everybody. I hope I find you and your family and friends safe and in good health this evening. I'm Craig McClain, Executive Director of the Louisiana University's Marine Consortium, aka LumCon. And I'd like to thank you for joining us tonight for LumCon's online science series. And every other Thursday night at 7 p.m. Central, we're inviting Louisiana scientists to share their research, and we'll give you the opportunity to listen and ask questions of some truly amazing scientists doing some truly extraordinary science. And I hope you will continue to join us each and every time we give one of these seminars to explore more of our world from the comfort of our homes. And you can find more information about the complete online lecture series at our website, lumcon.edu. Just scroll down on our homepage to the link under news and events. Well, tonight is my privilege to introduce colleague and friend and collaborator, Dr. Brian Roberts, who's the Associate Director of Science for LumCon. Brian was born in upstate New York and has had 41 different mailing addresses in life despite living in the same house here in Homa for over 13 years. That sounds pretty normal for an academic. He received his bachelor's in biology and philosophy from the College of William and Mary, received a master's degree from Boston University Marine Program when it was still based out of the Marine Biological Laboratory down in Woods Hole. received a PhD from Cornell in ecology and evolutionary biology, and was part of the interdepartmental biogeochemistry and environmental change program. And then he went on to complete a postdoctoral research associate position in the environmental sciences division at Oak Ridge National Laboratory. He joined the LUMCON faculty in August of 2007, became the associate director of science of LUMCON in 2017. And he continues to oversee the ecosystem ecology and biogeochemistry lab at LUMCON. Brian, so glad to have you here tonight with us, giving this lecture. Been very much looking forward to it.
All right. Thank you. Can you all see my screen? Yeah. Is that?
Yes, we can, Brian.
Okay. So we'll resume that. Okay. So today I want to talk about the complexity and connectivity of coastal ecosystems in the context of trying to understand what we've learned from the Deepwater Horizon oil spill. And as Dr. McLean introduced me, I'm the Associate Director of Science here at LUMCON. And the work I'm going to be talking about is the work that my group has been involved in for most of this last 10 years. But it's really not fair to say that this is a presentation that's only based upon what I've contributed. The Coastal Waters Consortium has been run by Nancy Rabelais, and we've had, for our different grants, anywhere between 21 and 26 principal investigators as part of that. I've led the biogeochemistry and microbial ecology and plant ecology components of that, along with Ann Bernhard of Connecticut College and Ann Giblin. I've had a number of research scientists and postdocs that have worked in the project, as well as a lot of different staff members that have come through my lab, graduate students, and then undergraduates as REU interns for the most part, as well as some local university students as undergrads. So there's been a lot of people that contributed to the work that we're going to be talking about today. I'm also going to be talking about, now that we're at the 10-year anniversary of the oil spill, we're making a lot of efforts to try to understand and summarize what it is that we've learned over these last 10 years. And as part of that, I've been asked to lead a wetland synthesis working group by the Gulf of Mexico Research Initiative that's funded a lot of the work that has to do with the oil spill. And I want to make sure I point out my co-leads in that effort, Scott Zengel, Ken Heck, Herb Mendelson, and Robin Zarabecki. We've had a number of participants that have been part of that working group, as well as we've had motivators from GOMRI. We brought in external industry and agency participants as well to contribute to that. So now that I've covered acknowledging people right up front, I'll get into what I'm going to talk about. So when you look out at a salt marsh, it may seem like it's a pretty simple system. Like in this particular view, you pretty much are seeing mostly one species of plant that's in front of you. But when you look closer, you see that these systems are actually quite complex. And so when you get down into the grass, the picture on the left over here is a picture that the marsh periwinkle snails are basically taking over the entire view. And if you look down on the marsh sediment, you see fiddler crabs are scurrying around. There's a lot of complexity to this. And if you actually look at a whole food web for a marsh and marsh associated species, things like dolphins don't go swim on the marsh, but they utilize a lot of the fish species that spend part of their life cycle on there. It's very complex, and there's a lot of connections. Different organisms have influences on a lot of other organisms. And so it can be a little bit overwhelming to try to understand how the system as a whole responds. And maybe natural to want to look at your own focal species or taxonomic group and try to understand what the oil spill means to that. But if you look at it in isolation, you may miss part of the story. So to help us illustrate this, I'm going to go through this conceptual model and see this beautiful watercolor in the background that I'm using here that was provided by Mert Conover. And so we can take all those individual species and taxonomic groups and kind of bin them into larger categories. So you have your marsh primary producers, you have invertebrates, things that fly, things that climb, things that graze along the surface, things that actually burrow into the sediment. And we have vertebrates that are on the marsh platform. We also have an aquatic interface. And then a lot of the processes that take place in a marsh, the cycling of carbon, the degradation of material, the cycling of nutrients, making them available, is mediated by microbes, and those processes are biogeochemistry that are influencing these. And what I've highlighted here in blue are some of the connections where you have changes in the marsh primary producers that influences invertebrates. It also influences the microbes, and it can influence the aquatic invertebrates as well. And they play a role in regulating how much erosion and how much sediment accretion takes place in a marsh. There are also a lot of external factors. There's physical forcings, the hydrology, the delivery of sediment and materials can influence the way that a given marsh functions and what species may be found there. There are also a number of other stressors that take place. So we have things that are changing with sea level rise, changes in salinity, changes in vegetation or invasive species, changes in our nutrients that are being delivered. These all can influence the way that a system functions. Now, we take the case study of oil. Oil itself has toxicity and physical impacts. It can have direct impacts of oil on any of those components within the system. But there also can be a lot of indirect impacts of oiling on the ecosystem as a whole. And if you look at where those indirect pathways are, those are pretty much overlay the pathways we talked about on how the different components are connected to begin with. So if you have oiling influences the plant productivity, that may have an influence on the things that normally eat plants, or they rely on the plants for providing oxygen to the subsurface soil environment. And those same physical forcings can have influences on the magnitude effects of oiling on the marsh ecosystem. So it can influence where we get oiling, how far the oiling extends into a marsh, how intense it is, as well as what other materials are being delivered to that system. So it's really taking an integrated approach to understanding how perturbations or disturbances influence the way systems function, is what we want to talk about today. So the case study we're going to talk about is the Deepwater Horizon oil spill, which took place over 10 years ago now. It's hard to believe. But when that rig exploded, it killed 11 people. It took 87 days before we were able to cap it. We released almost 5 million barrels of oil, not to mention a comparable amount of hydrocarbon gases into the ocean environment. We were able to collect almost 1 million barrels of oil, but that still left over $4 million barrels of oil that was moving towards the coast of the Gulf of Mexico. This picture over here on the upper left is actually showing the dark color, the black in here is actually showing the oil that's being entrained into Barataria Bay, into some of the areas that were heavily oiled that you may have heard people talking about in Bay Jimmy, Bay Baptiste areas of Barataria Bay. Coastal wetlands, including salt marshes, mangroves, and reed marshes, were all oiled. And it can be quite extensive. You can see the oil right here, tens of meters into the marsh platform. It can end up with a very extensive mousse that ends up onto the marsh platform and obviously can have big impacts from that. In total, there are over 2,000 kilometers or 1,300 miles of Gulf shoreline that was oiled as a result of the Deepwater Horizon oil spill. And over 1,100 of those kilometers, or almost 700 miles, of the shoreline that was oiled was wetlands. And about 95% of those oiled wetlands were in coastal Louisiana. And there are over 200 miles of marsh that received moderate to heavy oiling. Now, marsh shorelines, as you might expect, receive the majority of the heavy oiling. Reported range is that it extended anywhere from 0 to 100 meters is the largest that we've seen documented in terms of how far into a marsh oil may have gone. Most observations put it at anywhere between 10 and 20 meters within the edge. You can see this is in the upper right is a little bit more of a close-up of that. And here is a site that shows what oil extending into 40 meters into a marsh may have looked like. Now, Louisiana is an area that experiences very rapid land loss through shoreline erosion. But there is evidence that heavily oiled shorelines eroded faster than the lightly or unoiled shorelines. It's a little bit complex to be able to tease apart the cause of all of that, because those areas that receive lots of oil also tend to receive a lot more wave energy, so may be experiencing higher rates of loss to begin with. But there's clearly evidence that the heavy oiling did result in faster erosion or loss of marsh after the spill. So what I'm going to do here is I'm going to talk about a few of these different groups. And even though I'm an ecosystem ecologist and biogeochemist, I'm going to actually, in this spirit of time, actually skip over talking explicitly about the biogeochemical and microbial responses today to talk about a few of these different groups and what we've learned. And then I'm going to talk about an approach that we've taken here at LumCon to try to understand, to tease apart some of this complexity to understand the impacts of oil. Okay, so this is just another version of a map showing the extent of oiling. And one thing I want to point out is that these different areas of wetlands are often dominated by different vegetation types. Much of these interior marshes that are found throughout Louisiana are actually dominated by Spartina alterniflora, smooth cordgrass, or it's co-dominated with Juncus rhamarianus, a thrush. Excuse me. There are other species that are present in many of these marshes, but those tend to be the two dominant species that are there. And this is where most of the research has been focused over the last decade, or on these marshes. As you move closer to the Birdfoot Delta, the Mississippi River, when you're in this more freshwater environment, they tend to be dominated by Phragmites. And then Louisiana, like a lot of places, is seeing the northern expansion of black mangrove, Avicennia germinans, into our previously salt marsh environments. And these tend to be found closer to the southern parts of this region. In the area around Port Fourchon is one of our most extensive areas, but it is spreading throughout our region. And each of these has different sensitivities to oil that I'll talk about. So when we talk about terrestrial prime producers and what the impacts are, the overall impact and recovery following the Deepwater Horizon spill was dependent on the oiling intensity, the proximity to shoreline, the plant species, and how much the oil actually penetrated into the soil. When it penetrates in the soil, that has negative impacts on the below ground biomass, the root structure. It also changes the chemistry of the soil, which tends to influence the plants as well as the other inhabitants of the soil. So the lightly oiled shorelines typically showed minor vegetation impacts, and they recovered relatively quickly. Moderately oiled shorelines often showed more significant impacts, and that resulted in some slower recovery. The heavily oiled shorelines, of course, had the most severe impacts, with impacts generally decreasing with distance from the shoreline. So the further interior you are, the further removed from the heavy amounts of oil, the lesser impacts. Now, in areas where both species are present, Juncus typically was more impacted than Spartina at both the moderately and heavily oiled sites. This had important implications not only for the impacts in terms of the metrics, but of vegetation, but also the recovery. So many of the vegetation metrics took several years to recover. So we're talking about the stem height, the biomass, changes in their allometry, stem density. All of those things tended to take several years to respond, with recovery of juncus being significantly slower than alterniflora for most of these metrics. And the recovery of both species is often prevented along rapidly eroding shorelines. So if you're in an eroding shoreline that received heavy amounts of oil, it was often the case that the plants were not able to recover fast enough before the marsh eroded away. So that tend to be areas where we had extensive amounts of loss. So the other primary producers that I want to briefly talk about are those Phragmites dominated marshes. The oiling often killed the above ground tissue, but growth and recovery was actually relatively good. And vegetation cover was not significantly different from reference marshes, unless the oil penetrated into the soil and impacted the below ground tissues, in which case then we often would see some impacts on the Phragmites in the successive years after oiling. The Avicennia dominated wetlands, those patches that are adjacent to shorelines were clearly negatively impacted by oil, but we have very limited published data, and there's really no recovery information in terms of how long it's taken for that to recover. It just has not been studied very well. Another important primary producer on the marsh platform is the benthic microalgae. It's the algae that's growing on the sediment surface. It'll be growing on the bottom of plant stems. Often that's something that's a preferred food source for many components of the food web. They're clearly negatively impacted by oiling. They began to recover with the return of above ground vegetation. So often it's within about 2 years post spill, depending upon the severity oiling in a given location. When we talk about terrestrial invertebrates, we can talk about several different classes of those invertebrates. When we talk about the infauna, I'm going to talk about 4 today that are the ones that have received the most study to this point. We talk about the infauna, the abundance and diversity decrease initially. We often saw almost 100% mortality at the heavily oiled sites. Recovery of most taxa related to the recovery of plants. So as the Spartina alterniflora biomass return, as the benthic microalgae biomass return, then the infauna tended to be doing better and return. There are slow recovery of some species that related to the slow recovery of those plant biomass and soil properties. In general, the total abundances recovered in less than 5 years, but the community composition did not fully recover to the point at which studies have been completed, which is now 6.5, 7 years. At that point, we still were not seeing full recovery in the community composition. So we had the same total number of invertebrates that are present in the infauna community, but the composition of them was different. Moving above the sediment surface and looking at the terrestrial arthropods, these are your insects and your spiders. The total arthropod numbers and as well as across feeding guilds were definitely reduced by oil exposure, but they recovered relatively quickly. In most studies, it seems to show that that's within a year or so. The greenhead salt marsh horsefly is actually, as a larvae, is the top invertebrate predator in the marsh soil. Has proven to be a good bioindicator species. It has immediate impact of oil, and it had recovered by 5 or 6 years, and it followed a succession of plants and infauna. So looking at how it's responded and recovered gives you a good sense overall about how the overall system has been responding. One of the pictures I showed earlier on, you can see fiddler crabs scurrying across the marsh surface. They've been pretty well studied as well. Fiddler crabs at oiled sites were initially less abundant. They were also tended to be smaller, and we saw a shift in the species that was dominant of fiddler crabs that was dominant in those heavily oiled marsh sites. The recovery of species composition again, was related to the return of vegetation. So you can see the connectedness. So you need the plants to actually come back before you start to see several of these invertebrate communities to recover. And by the latest studies that have come out in the literature that tracked it for 5 years, they're still not seeing full recovery of those communities. And the other one of the species that I showed you, the marsh size view was the marsh periwinkle snail. Their densities and shell sizes were negatively impacted initially at oiled sites. Their densities have increased over time. They actually have reached somewhat higher densities at the heavily oiled sites. The average shell length at heavily oiled sites remain less than at the less impacted sites. And we're still seeing negative impacts 9 years post-spill. And those negative impacts were the strongest at the heavily oiled sites. So for all of these communities, there's been a delay in terms of being able to see complete recovery through the period in which we've studied them. So terrestrial vertebrates have been studied a lot less frequently than the invertebrates or the plants. Far and away, the most studied bird on the marsh is the seaside sparrow. They've experienced a range of toxic effects, physiological effects, metabolic effects. They've seen reduced reproductive success initially, but that was not detected after a few years. So the first couple of years after oil exposure, they saw reduced reproductive success, but that has returned. And there hasn't seemed to be any difference in the trophic position or what they're eating relative to oiled or unoiled sites. So their diet hasn't changed. In terms of other birds, there are early mortality rates in birds thought to be very high, but the error on these estimates is also very high. We have very little confidence in our ability to constrain exactly how high the overall bird mortality was as a result of the oil spill. There have been a series of experimental toxicological studies on birds, but there have been few studies on the impacts of the marsh resident bird populations. So, how many birds died on the marsh that utilize the marsh? We don't really have a good handle on that information, unfortunately. In terms of other terrestrial vertebrates, mammals are very common on the marsh. There have been relatively few studies that have actually focused on the impacts on mammals. Few of those studies have largely focused on marsh rice rats over here on the left, and their physiological or diet impacts on being exposed to an oiled site. Very little has been studied about raccoons. And reptiles, we know they're there, but there's little known about the impacts of oiling on reptiles at this point. Now, getting a little bit wetter into the aquatic vertebrates, we do know that oil has deleterious impacts on fish individuals. So there's general widespread agreement in the literature that oil alters the toxicity, development, behavior, reproduction. So this is on the right here are a variety of different fish species in their larval stage, and seeing what the deformities associated with oil exposure on the right compared to the control. And on the left is a red snapper, where you can see a pretty pronounced lesion on it as a result of oil exposure. While we've seen very strong impacts on the individual level, when it comes to looking at fish populations, and what this map here is a series of field studies that have taken place since the spill from Louisiana to Florida, and some of these have been in seagrass habitat, several have been in marshes, including some that we've been involved with here in Louisiana. And what we did is, this is Charlie Martin out of Florida led this component of this assessment, is sort of scored these based upon what their impact was. So if you got a 3 as a score, it meant that you actually saw no effect on the population after oiling. These blue values, these 2s, are showing that there's slight improvements. A red value, or a 4 or a 5, would be an actual decrease or an impact, negative impact on the population level of the fish communities, which we have not really been seeing. There have been some significant impacts on marsh-associated aquatic vertebrates. I showed you in that food web at the beginning, the dolphin at sort of the top of that food web. We've seen the death of over 1,100 dolphins between 2010 and 2014. And in the areas that had oil exposure, the dolphins generally have been in very poor health, having cases of lung disease, reproductive failure, adrenal gland and lung lesions, and then slow health recovery. So we have seen some very significant impacts on some components of the community, some of the top predators. So on first thought, this is a little bit perplexing, right? We know that we have these negative impacts are consistently detected on the individual or the organism level. Yet when we go to the population, we're not detecting those negative impacts. So in a review that Joel Fadri led a few years ago now, they came up with a variety of different potential mechanisms that may explain the absence of these population level effects despite organismal level responses. These can range from behavior to the difference between the toxicity level of what was used in these experiments, what we see in the field. And 2 I want to highlight here have to do with changes that we see in predation on the species, the target species of interest. So we already saw that dolphins are heavily impacted. So if you have an impact on the predators of a species, then that is going to be a release for the study organism. So that can happen both from natural predators as well as human predators in fishing. So one thing that happened after the oil spill is we shut down the fishing industry for a large period of time. And so as a result, you could see either no change in the target species, fish species, or even slight increases. That doesn't necessarily mean that the oil did not impact those populations, but they've had a release of predation pressure on them, so it may seem that way. Okay. So I've talked about a lot of different components here of how these ecosystems have responded. One thing that's challenging is that this is a multi-stressor environment. So there are a lot of things that can potentially impact different components in the overall ecosystem of these marshes. In addition to the ones that we've talked about, we've had difficulty detecting impacts and recovery from oiling because there are differences between sites that are oiled and unoiled prior to receiving the oil. There's something fundamentally different about a place hydrologically in terms of the materials that are delivered to it. That was one that received oil and one that did not. And so maybe you're not comparing apples to apples. The timing, spatial distribution, extent of oil is highly variable. There's differences in the degradation rates of oil that's on the sites. We lost habitat. We saw shoreline erosion was impacted. And over time, you see decreasing amounts of residual oil, so trying to tell how much of it is an impact of oil. And then we've also had these other instances. We've had re-oiling events. Hurricane Isaac came through in the fall of 2012, 2 years after Deepwater Horizon spill. And this is a picture up here of one of my marsh sites that was our reference non-oiled site that we were at 2 weeks before this. It didn't look like that. This is oil that has killed back this stretch of marsh right here. And when we actually had that oil analyzed, it had fingerprints that indicated that it was Macondo oil. This is likely oil that was on the shelf and then got picked up by the hurricane and brought onto the marsh. It's not an isolated incident. We know that there are several people have documented those sorts of impacts. That can also add to the complexity of looking at a recovery trajectory. So one approach is to try to do a large scale manipulation experiment. where the only thing that you're manipulating is the oil itself. So that's what we tried to do here at LumCon. We built these large-scale marsh mesocosms. These are 12 isolated Spartina alterniflora marshes that each have their own tidal surge tank that exchanges tides with them. And then those two, the surge tank and the marsh, are a hydrologically isolated unit. So once the water comes in there, it doesn't connect with any of the other systems. We completed planting these with Spartina. These were intact marsh chunks that were dug up and planted. So these were not seedlings that were put in January of 2018. We did baseline measurements that spring and summer. We moved water via air blowers for each tidal cycle. And now we're going to try to show you some drone footage. We had a little trouble with this earlier, so you can get a better perspective about how these look. So let me see if this will work. Okay. I hope you can see this. Okay, so let me pause it. So this is the pump house behind LumCon, and in here we actually pump water up from the bayou, and this is right next to our environmental monitoring station, so we know what the conditions are of the water in the bayou at all times. We actually pump it. We pump it through this pipe. There's actually two pipes there. I'm not sure why our video is doing that. Okay, I'm gonna pause it there. So the water comes up through these pipes and it comes into this mesocosm facility. The water then comes directly into these small tanks that are covered here with mesh. Those are our tidal surge tanks. Then we move water back and forth via air blowers and a series of air lifts. So we can raise the tide, moving water into the marsh. We can lower it back into the tidal surge tank. And then the water runs out. We'll do an exchange of water. So that way our residence time is about 45 days, which the residence time of the bayou is. And we send it down to our treatment center at the other end. I'll try to switch and show you a different video that I think will give a little bit better. Okay, so the overall system is about 140 feet long. Each one of these mesocosms is a 10-foot diameter marsh. They have a trough on the outside. So water comes in from the outside and floods from the outside edges and over the top, and then it comes down the same way. It does allow lateral flow through the sides. We have a series of holes in the trough that allows the water to come in and out. These are a large enough scale in which we've done a series of experiments where we've had marsh fish involved in the process and looked at some of the impacts of oiling on that. We moved intact sections of marsh, so that came with all of the invertebrates in the soil. We didn't have to bring anything in, it was all already there. We've been able to track that over time. On this far end, here is our treatment system. So when we have the receding tide, we pull water out, we bring it in through equilibration tank, we fill it through a bioreactor, then it has a polishing tank, and then the water comes back out to the bayou after all the oil has been cleaned from the system. So this is just some footage to give you an idea about the scale. Those are members of our research team doing sampling. At this point, I think they're doing random quadrat sampling of the marsh. Hopefully you're back to seeing this. I'm happy to take questions about how the system works. If anybody wants to find out more about it, I'm happy to talk more about it. It's been a very long process to get this built, designed, then built, and then to be able to do these experiments. So one of the things that we wanted to be able to do is to be able to isolate what the impact of oil is from all these other stressors. So we designed a large-scale experimental oil manipulation experiment in which, after the plants had been in the marsh mesocosms for a year and a half, and we had very good baseline data, last July, we oiled them. So we applied Louisiana light sweet crude oil that was evaporatively weathered, about 25% by weight, comparable to what we saw in terms of the oil that made it on shore. It's designed to be in oiling levels that are at the concentrations comparable to what we're seeing in those different categories that showed up in those oiling maps that I showed you earlier. The low, moderate, and high oil. We basically divided the marsh into grids, and we applied a certain amount of oil via syringe into each grid at high tide, brought the water down, moved it up and down in contact with the surface to maximize the contact before we resumed regular tides later on. I'm going to show you just a few highlights of some of the impacts that we've had and we've seen here. This is some pictures of a control, a low oil, a moderate, and a high oil tank 1 month post oiling. At that point, you can see an increase in the amount of dead tissue, a lot of yellowing tissue that's found, and especially in the high oil system. You take a look at, just to make it easier, just to have the control on the high. This is 3 months post oiling, which you can see a very different looking marsh. Now, we didn't go for a level of oiling that would just completely kill everything. We wanted to have sort of a realistic high oil concentration event, but it wasn't one that completely decimated the plants. So just to show you a few of these responses, and I was trying really well up until here at the end about not showing any actual data, but I kind of ran out of time putting that part together. So these graphs are all going to be the same. So once I explain them once, it shouldn't be that difficult to follow. So the control are all the white symbols. The low are the yellow, blue is moderate, and red is high. Time is on the x-axis. At first, we're looking here at live stems. The white area is before oiling, and the transition is the date of oiling. So the take-home message here is that the live stems are lowest in the high oil treatment throughout the entire first 4 months. This is through basically the end of last growing season. And we saw decreases in the moderate to low by the time we got 4 months in. Live leaves, so the number of live leaves on the plants also was lower in the high oil treatments. We saw a lot less live. We also see more dead. We have more dead stems in the high oil treatments by the time we got 2 months in, and that the moderate were higher by the time we got 4 months in. Now, importantly, for the continuation of growth of the system, one of the things that we saw is that we saw less new growth. These are really short stems, things that are less than 10 centimeters high. Almost immediately, we saw less new growth. The red circles are way below all the others, and they maintained that throughout these first 4 months. And by the time we got to these last couple months of last fall, the low and the moderate also were observed to be lower. So overall, the plants were clearly negatively impacted. I'm going to show you just, and the last thing about plants I forgot was that during the period in which we have peak flowering, we actually saw a slight increase in the flowers in the oil treatments. And the question is whether or not this is a stress response to actually put more of the resources into producing flowers or not. And I didn't get to show you this, but one thing that we do here is that we actually have intact cores that we're able to take out of the marsh, and we bring them up to a radiation lab, and we actually scan them on a CT scanner, and we can assess the below ground biomass in that core and then put it back in the marsh, and then we can resample it. And it appears that we're seeing a slight decrease in the below ground biomass after oiling. We also measure gas fluxes. These are chambers here, a light chamber and a dark chamber, to measure gas fluxes, to look at how much carbon is sequestered by the plants. Now, this is normally plants take up carbon dioxide from the atmosphere through photosynthesis. So a less negative value is actually a decrease in that rate. And what you can see here is that these red symbols are much, much lower in rate than the other symbols. So high oil treatments had prolonged decrease in gross CO2 uptake. And this decrease is driven by the fact we have less live leaves. If you actually scale it to the rate per leaf, it's actually not different. And we saw a small transient decrease in the moderate oil, but we saw no differences in the low or the control. In terms of methane emissions, which is an important greenhouse gas for warming, we see a transient response in the high over the first couple of months, but we don't see any difference in either the moderate or the low. And I couldn't have a talk, didn't have anything to do with the biogeochemistry, so I'm just going to show you a little bit about one of the important processes in the nitrogen cycle, the process of nitrification. And the important thing to notice here is that there's no differences between any of the mesocosms for the 1 year before we oiled. There is a trend towards an increase as you get later into the summer. After oiling, there was clearly impacts immediately after oiling, but the responses were not the same across the oil level treatments. And now I'm going to zoom in on this so you can actually see it a little bit better and talk about each one. If you look at the red, the nitrification rates in the high oil treatment dropped to 0 within the first week. We could not detect it anymore. Basically, nitrification, which is an important process in making nitrogen available in the system, is basically shut off, and it maintained that way for at least the first 3 months. We see that the blue symbols here, the moderate oil, were suppressed, and they remain suppressed for at least those 3 months. But we saw no difference, maybe a slight decrease at the first month, but really no difference from low. That's kind of what we've seen for a lot of things. High oil is a heavy impact, moderate somewhere in the middle, and low often hasn't done very significant impacts. So those are just a few of the types of things that we're doing in these mesocosms. And I hope that I've given you somewhat of a feeling for the complexity of trying to address questions about, what was the impact of oiling on an ecosystem, and that the advantages of taking a holistic look, looking at multiple components of the system and seeing how they respond and how the response of one component may influence the recovery of another, and ways that we can use large-scale manipulative experiments to actually isolate what the true impact of oil is amongst this multitude of stressors that we experience out in the natural world. And with that, I want to thank you for listening today, and I'll take any questions.
Thank you, Brian. That was excellent. And whoever your drone pilot is wonderful. I'm just kidding.
Yeah, I don't know. I think it was the same person that drew that watercolor. Watercolor, maybe.
We do have some questions coming in. So while our audience is busy typing those, we do have a lot of thank yous for the presentation coming in from our audience. So it seems like you have some fans out there. So the first question is, once you are done with the oil experiments, can the mesocosms be used for anything else?
Yes, the design is that we can do that. It's honestly, it's going to be a major undertaking. So, I mean, when we build these, so there is a 6-inch layer of gravel at the bottom of these with a French drain in them, so we can actually allow vertical flow of water if we need it. Then there is 2 dump trucks full of sand that is spread across those. And then we move 53 tons of marsh channel mud. And then we have 1,600 buckets, 5-gallon buckets full of intact marsh. So the actual physical removal is going to be a major undertaking. But after you actually treat the walls of the system, it could be used for something else.
Perfect. We have a question from Keegan. Hey, Keegan, good to see you. So Keegan's question is, what method did you use to select which mesocosm received what oil treatment?
So there's 2 parts of it. So we used a randomized block design. And the reason what that means is that we randomly assign tanks, but we recognize that at a system that's 150 feet long, that's also along the span of the dominant wind direction, that you may have some influence or variation as you move from one end of the system to the other. You're also moving a little bit further away from your air blowers, so we wanted to try to control for that. So each set of 4 is a block. So the 4 that are to the south are a block, the middle and then the north are a block. And out of those, then they were randomly assigned.
Perfect. Sorry, we have a lot of questions flying in. Here's one from Chandler. Thanks for the question, Chandler. Chandler wants to know, how has Hurricane Laura affected the mesocosm experiment, or is it too early to tell?
Hurricane Laura is not going to have impacted this experiment. And the reason for that is that the mesocosms are, the top of them is, well, they're 5 feet off the ground at that spot, which is not one of the lowest points on the property. So Laura, while it had 18-foot surges over in the western part of Louisiana, it actually only got within a foot of the top of the mesocosms. So it itself didn't really have anything. We just got wind that you would get if you're out in the marsh anyways. Hurricane Barry last year, however, did overtop the entire thing. And that was more of a logistical problem than it was anything else. I mean, it wiped out my entire computer control system. So we had to run lots of electrical conduit. It was not that long after we had oiled, so there was some dilution of some of the oil before it had fully adhered to the plants. But we know there was no cross-contamination. So a major flooding event that's high enough where it overtops the whole thing has some potential to impact, but you got to remember that as long as you don't end up with like a high oil treatment dumping oil into a control, the marshes that are out there right next to this are experiencing more flooding than we're seeing in the mesocosms. So unless it has a destruction to the system, it's not going to have a major impact on the experiment.
Great. The next question is from Tristan. Tristan is asking, given the unlimited funding, what kind of research would you like to see done regarding the impact of the oil spill? Or were there any studies related to the oil spill you wish had been done that can no longer be done? Maybe because of the time since the oil spill.
Well, that's what I'm guessing. I do think that one of the greatest challenges that many of us have had with the oil spill was the fact that even though there was a lot of research dollars that went into studying this, by the time many of us got to start doing our field studies, we were studying sites that were 2 years removed from the initial exposure of oil. So you miss some of that initial response. If you could have had pre-data, and that brings up some of those difficulties I pointed out about why having these controlled experiments are really useful, is that you don't necessarily know that that site that was oiled and the one that wasn't oiled actually behaved similarly before the oil happened. So that's a big challenge. I think that while there were a lot of large projects, I think that sometimes things were not quite as integrated as we talked about here, in terms of trying to look at all the different components. So I do think that that would be the case. I mean, it's hard to say you want to do something like that, but you know, these mesocosms are great, but there's still limitations of those. I mean, if you really want to do the best experiment, you probably should do a large scale field level oiling experiment where you have everybody lined up to sample everything that you need to from the beginning, sample before as well as after. I think we did a good job of doing a lot of things after, but we're guessing a lot. There's a lot of really field studies that conclude that there wasn't a large impact, but they all acknowledge it's like, yeah, but we started sampling 2 years after. Maybe we missed when that impact happened. So I think that's probably a way that you could do it.
Perfect. Megan has a question about the net around the tanks. Can you explain why there's a big net around the tanks?
Okay, well, so yeah, it took a long time to figure out how to get the right net, and then it took a long time to figure out how to get it up over there. So it's a large aviary net, and the reason for that is that it allows things like sparrows can fly in and out. We have a lot invested in this experiment, and you've got a 3 meter or 10-foot diameter marsh. You get one heron that decides to land in there and gets really annoyed and starts ripping something out, or you get a raccoon to go in there and just tear something up. I mean, you could basically destroy the whole experiment. So the idea is to try to keep out those really large predators that are having a disproportionate impact on that controlled experimental system.
Perfect. And do you have time for one more question? I know we're pushing the 8 o'clock hour. This one is going to be funny for you because we talked about this while you were making your presentation. So Arturo actually has a question about biogeochemical processes. Mm-hmm. So, you know, maybe throw some microbial information in there for Arturo.
Is there a specific question or just?
No, he just wants if you can just briefly discuss some of the oil impacts in regards to the biogeochemical processes.
Okay, so yeah, I mean, and I ended up touching on some of it a little bit in the end, talking about the mesocosms, in which we did talk about some of the nitrogen cycle as well as the gas fluxes, which are of course driven by respiration and other processes that contribute to CO2 release. In general, you know, there's impacts on the microbial community, changes in response to being exposed to oil. Those microbial community, certain groups tend to actually become more prevalent. Obviously, the oil degraders do. So you have an increase in them. You see increase in sulfate reducers that are often present there. Those will change the oxygen levels in the soil, which will then change a lot of the other processes that are associated with things. And those impacts that if they then affect things like the nitrogen cycle, which we all need nutrients to grow. then changes the availability of those that may influence how well the plants grow versus how well some of the fauna grow. So it's a little bit of a flair for it. That's one thing. I'm more than happy to answer more specific questions afterwards, but I also don't want to, bore everybody else to death with biogeo, blah, blah, blah. Speak.
Okay, I think that's the rest of the question. You guys, if I left your question just kind of hanging out there, and you really want or need the answer to your questions, feel free to email those to me. You now have my email. It should have come at least with the confirmation of your registration or your reminder email. You can send me those questions and I will get them to Brian and he'll get you those answers. So again, sorry if I did not get to your question this evening, but I really hope if you want or need those answers, you'll seriously take our,
I'm happy to answer questions.
Yeah. With that, I want to thank Brian so much for giving tonight's presentation. It was awesome. You're getting a lot of great feedback from people, and I'll share some of those with you. And I really want to thank our audience for joining us again tonight. You guys are awesome, always engaging, always have great questions, and we certainly love seeing you every Thursday. So with that, keep in mind that not next Thursday, but the Thursday after, we have another LumCon Science Talk with Dr. Tim McLean from Tulane University. So please mark your calendars and be ready for that talk information coming very soon. Good night, everybody. Take care of each other and take care of yourselves. Good night, Brian. Thank you so much.
Thank you.